All right, let's get started. So this is 3.1, the first video of Unit 3, talking about the thymus, its structure, and its function. So what's the breakdown here? Well, let's talk about Unit 3 in general. We're concerned with T cells. T cells, we're talking about where they come from. We'll talk about their development and the different classes of T cells. Your T cells are also excellent at recognizing the self-antigen, MHC, determining self from non-self. So we'll talk about that whole process as well. That deserves to be in the conversation in this unit. We'll also talk about viral infection and viral evasion, as T cells are heavily involved in that process as well. All right, but today is going to be pretty calm. We just want to give you the anatomy, the geography, where these T cells are being trained. And that is going to be the thymus. That is one of our primary lymphoid organs, and it is the site of immunological education of the T cells. It's surrounded by a capsule and a specialized blood thymus barrier. While these guys are developing, we want to be selective of what they are introduced to and what they are not. Very active in childhood, but it undergoes involution. Involution after puberty. Involution is a fancy word for replaced with fat and connective tissue. So I used to think of it like as opposite of evolve. It kind of just gets replaced with nothingness. You still have a little bit of thymic cells there, but it does diminish greatly after puberty. So there are some implications there. Maybe your thymic education is not so good in adulthood, and that's the case. Where do we find it? So we have the lungs right here, heart right here. The thymus is sitting right in the middle of that mediastinum, right behind your sternum. Okie dokie. The gross structure, by that I mean the zoomed out structure, big old dense bilobed organ here surrounded by a connective tissue capsule. That's in this light orange here. And it consists, each lobe has an outer cortex and an inner medulla. The outer cortex is the orange here, the dark orange. And the inner medulla is the purple. So why am I making a stink about those two regions? Well, different things are going to occur or are going to occur in those regions, and we need to be able to differentiate those. So the cortex, that outer region, that orange region, very cellular here, very dense, a lot of action going on. This is where all those T cells are coming in from the bone marrow. We call those developing T cells thymocytes. That's just a general term for those T cells that have just arrived. We have like in last unit with the B cells, they had their different pro-B and pre-B stage names. Your T cells will also have different names for the stages that they're in. But the broad term, we're just going to call them thymocytes for now. The developing video will get more into the different stages. You also have stromal cells that are going to provide assistance to these developing T cells. Stromal just means the cells of the tissue itself. So the cells that make up the thymus are going to help the T cells that make their way inside. In the cortex, we have cortical thymic epithelial cells. That is the stromal cell of the cortex. These are stellate in nature, so they look like stars. They have like big long arms. And they secrete thymic hormones that influence thymocyte availability and activity. Basically, they're going to help dictate what the thymocytes do once they get into the cortex. You also have macrophages hanging out in here. They're going to clean up any lymphocytes, any T cells, or pre-T cells that didn't make the cut in selection. So making sure that we don't have, dead debris hanging around. And this is where this blood thymus barrier concept that I brought up here comes into play. In your cortex, that's where all those T cells are just arriving. we don't want to worry about activating them, especially in their novice stage. So while we are building T cell receptors and while they are learning, we are separated from the blood here to allow for a quote unquote sterile environment for them to develop in. Then we have the medulla. Again, the purple region, the inner, the nougat. This is going to be less dense with cells, and the primary stromal cell here is the medullary thymic epithelial cell. These are a bit bigger, and they're loosely spread around. Remember, fewer cells is less dense in here. And again, they're going to do the same type of thing. They're going to coach and herd the thymocytes, the developing T cells, on where and what to do. You have a specified special version of the medullary thymic cells. Sometimes there's a class of them that forms this thing called the Hassel corpuscle. And basically, those medullary thymic epithelial cells, they kind of form, see if I can make a nice one. It kind of looks like a rose. So they just kind of circle around each other. Let's see, maybe I can get some leaves in here to make it look nice. Oh, that's beautiful. So obviously there's no leaves or a stem, but these medullary thymic epithelial cells kind of lap around each other, and they form this corpuscle here. And it's commonly called a rose whenever you look at it under a microscope. So this is just one of the textbook region or identifying factors of a thymus. Within the medulla, you have these little pockets here that look like roses. So that's number one identifier for if we're in the thymus or not, if these are present. So concentric, I'm sorry about that. There we go. Okay, come on, lock in. There we go, concentric MTEX, concentric medullary thymic epithelial cells hanging out here. So that's what a Hassel corpuscle is. Make sure you know that. So the vascular supply, a little bit different here. Same like artery is going to supply and same vein is going to drain, but it's more permeable here. There's no blood-thymus barrier. At this point, we want to test if these T cell receptors are working properly. So we have a little more exposure to circulating self-antigens. Right, that's important. We don't want to release any autoreactive T cells. So that's my medulla, and that's my cortex. So make sure you know these two cells that make that up, because they'll come up in next lecture for sure. All right, a little more here. I'm not going to make you guys know all the layers by any means, but the blood-thymus barrier is a cool concept because, again, it's prominent in that cortex as those T cells are coming in. It isolates those developing lymphocytes. We don't need them. We don't need them to activate right now. You know, like we don't need a first-year med student going into a cardiothoracic surgery. That'd be kind of crazy. They're not ready for that yet. So let's let them train up before we activate them. So we don't want premature activation. Again, you don't need to know these layers, but I'll just explain what they mean. So you have the endothelium of the blood vessel. Then you have the connective tissue, or you have the basement membrane and the connective tissue of that blood vessel. Then you have the basement membrane of the thymic cells, and then you have just the thymic cell themselves. So it's basically just all the connective tissue and cells of the thymus separating the blood from the environment that's within the thymic cortex. So we have this isolated, sterile region for these guys to develop without premature activation. I don't need you guys to know this stuff here. What I do need is your idea, making sure you have a good idea of involution. Okay, your thymus is not going to be that persistent post-puberty, and especially like in later years of life. Involution begins at this stage. And what this means is your parenchyma gradually reduces and is replaced by adipose and fibrous tissue. What is parenchyma? That is a good bio word to know, so I'm going to define it. It is the constituent cells or the functional cells of that organ. So the cells carrying out that organ's function. So in the thymus, it's the stromal cells, the C-tex and M-tex and the thymocytes. In a liver, it's a hepatocyte. In a kidney, it's a nephron. It's the functional cell of that organ. And in this case, they're just getting replaced by adipose, by fat and connective tissue here. I don't know why this happens. I don't have the evolutionary explanation for you. But that means T-cell college goes away right around the time you're in college, if you're on the traditional path, that is. So ERC, these epithelial reticular cell networks, they're still around. We don't completely lose them, but they do become sparse, a little more diffuse. So what does that mean? Well, if I don't have my functional cells of that organ, if my parenchyma is decreased, well, then my functional output of naive T cells is going to decline. So the thymic tissue still remains active, but it is declining in function as we age, and you don't even need to be that old. I mean, puberty is relatively early in life, so that is an early time for a sort of immuno-declining state to occur. Really interesting stuff. So we'll talk about that throughout this unit and the implications of thymic involution. But I need you to understand that concept. We start to lose that tissue as we age. Then a cool clinical correlate. You have something called DeGeorge syndrome. And this is the fancy way to say this is congenital thymic aplasia. Congenital means during formation, and aplasia means it didn't form. So you just don't have a thymus. So that means T cells can't go to college, they can't mature, they can't learn how to be T cells, and you have crazy immunodeficiency. You basically have no viral defense whatsoever. So very dangerous condition to think about. So that, not complex there. You literally just don't have a thymus. So in folks like that, we'll talk about throughout this unit. If you don't have a thymus, you can't make a functional T cell, so that whole branch of defense is knocked out. All right. So that's all I have for you guys today. Just understanding what T cell college looks like, where it is, and the different regions within. All right. So make sure you know those cells. Make sure you understand the map, because I think that will help you understand as we talk about the complex process of T cell development in the next video. Thank you very much for your continued attention. Have a wonderful day.